This lecture is on Chapter 12, the fundamentals of the nervous system and nervous tissue. All right, so what is the nervous system? The nervous system is the master controlling and communicating system of the body. It includes neurons and supporting cells, and nerves are actually bundles of neurons. There are three major functions of the nervous system: sensory input, integration, and motor output. This illustration shows the three functions of the nervous system. As you can see, sensory input carries information towards the brain and spinal cord. Integration occurs within the brain and the spinal cord, and then motor output carries information away from the brain and the spinal cord. There are two major divisions of the nervous system: the central nervous system, abbreviated CNS, and the peripheral nervous system, abbreviated PNS. The peripheral nervous system is further divided into two divisions: the sensory division, also known as the afferent division because it carries information towards the brain and spinal cord, and then the motor division, also known as the efferent division, because it carries information away from the brain and the spinal cord. Within the motor division, there are two systems: the somatic nervous system and the autonomic nervous system. The somatic nervous system innervates skeletal muscles, and the autonomic system innervates the cardiac muscles, smooth muscles, and glands. This illustration shows the layout of the nervous system. The central nervous system is the brain and the spinal cord, and notice that the function of the central nervous system is for integration, and it also has the major control centers. The peripheral nervous system is going to contain the cranial nerves and the spinal nerves, and it forms the communication lines between the central nervous system and the rest of the body. So let's look at the sensory division first, and it's colored in blue, and it's the afferent division, which means that it's approaching the central nervous system. I always remember A for approach. So the afferent division carries information towards the central nervous system, and it includes the somatic and visceral sensory nerve fibers, conducts impulses from the receptors in the somatic and visceral nerve fibers to the central nervous system. You can see that the somatic sensory fibers are located in the skin, and the visceral sensory fibers are located in the organs. The motor division of the peripheral nervous system contains motor nerve fibers that conduct impulses from the central nervous system to the effectors, which are muscles and glands. Motor is efferent, and I always remember E for exit. So it exits the central nervous system, carrying impulses to the muscles and glands. So the two divisions of the motor division are the somatic nervous system and the autonomic nervous system. The somatic nervous system controls voluntary skeletal muscles. The autonomic nervous system is going to control the visceral motor or involuntary of the cardiac muscles, smooth muscles, and glands. Somatic is going to be things that we are consciously aware of generally. So notice that somatic sensory is your skin, and the somatic motor is your skeletal muscles. So both of these are voluntary and you're aware of them consciously. So generally, somatic is going to mean that we're consciously aware of them, whereas visceral or autonomic means that you're subconsciously unaware of their happenings. Like, you don't have to notice them for them to do their work. And so notice that with the visceral sensations, you're not necessarily aware of your organs unless there's pain and distension and bloating. And as far as the motor, you're not really aware of your cardiac muscles, smooth muscles, and glands. They're just going about their business subconsciously. All right, so with the autonomic nervous system, that's further divided into the sympathetic and parasympathetic divisions. The sympathetic division is that famous fight or flight, and so it mobilizes the body systems during activity. And the parasympathetic division is resting and digesting. It's conserving energy and promoting housekeeping functions during rest. So looking at these illustrations, you can see that in the red from the spinal cord going towards the skeletal muscle are the motor fibers of the somatic nervous system. Running from the spinal cord towards the heart, it's showing a sympathetic motor fiber of the autonomic nervous system. And there are parasympathetic. The vagus nerve is going to innervate and keep the heart beating properly during parasympathetic housekeeping duties. So these could be either. And then same for the bladder. It's showing parasympathetic motor fiber here, but what that's going to do is that's actually going to cause the bladder to be able to empty and void. When you're under housekeeping, you can urinate, no problem. But with sympathetic, it's generally going to cause the bladder to, I believe, relax so that you're not thinking about going to the restroom. So if you ever notice you're really stressed out, trying to get a deadline reached, you're not necessarily noticing your bladder, right? So that's sympathetic. So these, in summary, can be both sympathetic or parasympathetic, and they generally are antagonistic to each other. The sympathetic division is usually going to, do one role, and the parasympathetic will do the opposite, and that maintains this homeostatic balance within the body. All right, so let's look at the histology of the nervous system. There's neurons, which are the functional cell of the nervous system that conducts impulses. And then there are supporting cells, which are called neuroglia or glial cells. And glial kind of translates as glue. And those include 4 in the central nervous system and 2 in the peripheral nervous system. We'll look at neurons first, and then we'll look at all of those supporting cells. As far as the special characteristics of neurons go, they have longevity. They can live, your entire lifespan. However, they are amitotic, much like we saw with skeletal muscles. So even though they'll live for a long time, if they're damaged or dying, they tend to not regenerate, which of course science is finding out more and more about that every day. And they also have a high metabolic rate. They require extensive amounts of glucose as their primary energy source, and they also need a continuous supply of oxygen. Neurons come in many shapes and sizes, but generally consist of a cell body connected to one or more processes. The cell body consists of the nucleus and cytoplasmic organelles. Processes extend from the cell body. These include dendrites and axons. Dendrites are short and conduct impulses towards the cell body. The single axon is long and conducts impulses away from the cell body. The axon originates at the axon hillock, and that's where the action potential is going to be propagated. And then myelinated axons can contain nodes of Ranvier, which will propagate the signal or action potential away from the cell body. This illustration shows a neuron, and I meant to get that out of here. I've been going through and making sure all figures are not labeled by chapters because the chapter differs in the different books. But with your OpenStax book, this is chapter 12. All right, anyway, I digress. The dendrites here are going to be bringing the information towards the cell body. And this is the axon hillock. Hillock means like a hill or a promontory. And that's where the action potential will be propagated. And then once it fires, it will hop along the nodes of Ranvier in a process called saltatory conduction as the impulse travels from the axon hillock towards the axon terminals. And where the axon terminals join to either another neuron, a gland, or a muscle, that's going to be a synapse. Neurons are classified based on their structure and their function. Neuron structure refers to the number of processes extending from the cell body. Neuron function refers to whether the neuron receives sensory input, performs integration, or conducts motor output. And there are three structural classes of neurons, each with specific functions. Unipolar neurons have one process sticking off the cell body, and they are sensory. Bipolar neurons have two processes sticking off the cell body, and they are sensory. Multipolar neurons have many processes sticking off the cell body, and they are either integration neurons or motor neurons. So unipolar neurons have a cell body that sits on a small process connected to a long axon. Unipolar neurons make up the majority of sensory neurons of the peripheral nervous system. Unipolar neurons include somatic and visceral sensory neurons, and they're found in both cranial and spinal nerves. The cell bodies of unipolar neurons are found in ganglia, which are swellings on the neurons or the nerves. And here with the unipolar neuron, the dendrites will be where the receptors are located, and the long axon has two divisions there, a peripheral process that will lead to the ganglion, and then the central process, which will carry that information into the spinal cord. Bipolar neurons have one dendrite and one axon connected to the cell body, and they are rare and are found in the retina of the eyes and the epithelia of the nose. So even though they're rare, they're extremely important because they provide the sensory input for sight and smell. So here we can see the dendrites with the receptors that would be found within the retina traveling towards that cell body. And then the axon with the axon terminals will be then synapsing in ganglia and in the optic nerve of the brain, basically. Multipolar neurons have many dendrites and a single axon connecting to the cell body. They are the most common type of neuron, and there's two major types of multipolar neurons. As I mentioned, there's integrating multipolar neurons and motor multipolar neurons. So the interneurons are found in the central nervous system, and those are the integration neurons. 99% of neurons are interneurons, and so that shows you how much of the neurons are compacted into the brain and spinal cord. And then motor neurons of the peripheral nervous system conduct motor signals to the muscles and glands. Let me move this illustration out of the way. So they're motor neurons of the peripheral nervous system conduct motor signals to the muscles and glands, and they include the somatic and autonomic motor neurons. Recall that the somatic motor neurons are going to travel to the skeletal muscles, and then autonomic motor neurons will travel to cardiac muscle, smooth muscle, and glands. Neuroglial cells are supporting cells of the nervous tissue. They outnumber neurons, believe it or not. They do not transmit nerve signals. They are smaller than neurons, and they can divide by mitosis, so there is regeneration occurring there. Neuroglia cells occur in the central nervous system and peripheral nervous system. In the central nervous system, they are the astrocytes, microglial cells, ependymal cells, and oligodendrocytes. In the peripheral nervous system, they are satellite cells and neurolemmacytes. So this is an astrocyte. It's the most abundant neuroglial cell, and the astrocyte is going to form what's called the blood-brain barrier, where it will have feet that will connect to the capillary, absorbing the necessary nutrients from the bloodstream, and then transmitting it into the cerebral spinal fluid and also facilitating the neurons' nourishment as well. It will also guide the formation of synapses between neurons, and if a neuron is damaged, astrocytes will fill in the space left behind. Microglial cells are the defensive cells in the central nervous system, and they're very small. That's what micro means, and they are going to act like the macrophages and the white blood cells, because with that blood-brain barrier, we're not going to get white blood cells traveling into the central nervous system. So the microglial cells will play that role. And they can be implicated in some disorders if they go awry. But, you know, any cell that goes awry is going to be implicated in something. But you might see, you know, them mentioned in like multiple sclerosis and stuff. All right, then there's the ependymal cells. These are very similar to epithelial tissue. And in some places, the books will actually describe them as a modified epithelium. And they have that basal layer that is going to be in the brain or spinal cord tissue. And then they have the apical layer that will be next to the cerebral spinal fluid. And they are what actually produce and then circulate cerebral spinal fluid. And of course, cerebral spinal fluid is mostly water. It will contain ions and the necessary nourishment for the neurons and other neuroglial cells. Cerebral spinal fluid also provides buoyancy to the brain and spinal cord, helping to decrease the pressures put on it. Then the oligodendrocytes. Oligo means scanty or few. Dendro means branching. So these are going to be neuroglial cells that have processes that can branch off of the cell body of the oligodendrocyte, and they will wrap around axons, forming myelin sheaths. And myelin sheaths are insulators. The electricity cannot pass out of them, and so it basically keeps the electricity traveling down the axons of those neurons and helps to facilitate nerve impulses. These are the cells that are affected in multiple sclerosis. So as I mentioned earlier, microglial cells can sometimes be implicated in diseases such as multiple sclerosis, which is, you know, an autoimmune disease. And so microglial cells may, in theory, attack oligodendrocytes and break them down, causing the symptoms of multiple sclerosis. So research is ongoing. All right, now we're in the peripheral nervous system. So satellite cells are going to be shown around the cell body of the neuron here, and they're going to perform similarly like the astrocytes. And then the swan cells, also called neurolemmacytes, are going to surround the neurons in the peripheral nervous system. And the neurolemmacytes or swan cells are going to be forming myelin sheath, just like the oligodendrocytes do. And notice that it's a little different looking. It looks like beads, and there'll be little exposed areas of axons in between the myelin. And so what happens is the electrical impulse will travel through the myelinated region because, you know, it can't be conducted outward, so it'll focus it. And then right at those nodes of rim via, the action potential will be repropagated because there's going to be voltage-gated channels that will reinitiate the action potential. So it kind of hops down the neuron axon, and it speeds up that conduction in these very, very long nerve fibers. So that's why they're in that shape in the peripheral nervous system versus the way that the oligodendrocytes look in the central nervous system. I guess it just has to do with like larger, more powerful, longer axons and the ability to carry that impulse many, many meters. And so there's the anatomy of a neuron once more to just kind of bring you back to the dendrite cell body, axon hillock, and then the axon itself, nodes of rim via, axon terminals where the synapse would be. And then now you recognize that there's the neurolemmacyte and the swan cell making up that myelin sheath to help conduct that impulse faster. So, speaking of nerve impulses, let's talk about neuron physiology. Both the neurons and the muscle fibers are excitable. We spent time talking about excitation, contractional coupling in the muscle chapter, and the first step in that is excitation of the muscle fiber by a neuron. And so in order for a neuron to send that signal to excite the muscle fiber, the neuron itself must be excitable and undergo action potentials. And so the electrical energy is conduction through the movement of ions across the cell membrane. And here are the terms that are associated with this electrical energy. The voltage is the separation of electrical charges across the cell membrane, and it reflects the distribution of oppositely charged ions. Current is the movement of ions across the cell membrane, and this is the electrical energy harnessed to actually do the work. So when that action potential is propagated, that's the current that flows. Resistance is the opposition to the movement of ions across the cell membrane. The cell membrane will provide resistance to the movement of the ions, and resistance will decrease when protein channels open, which is why all of those voltage-gated channels and even leakage channels are so important. So let's talk about those channels. Several types of protein channels facilitate the diffusion of ions across the cell membrane, and they include leakage channels, which remain open, and that allows ions to diffuse freely, and that's what kind of forms the membrane potential at rest. And then there are the sodium-potassium pump, which kind of further establishes that resting membrane potential. Then we have gated channels. So we've got mechanical gated, chemical gated, and voltage gated channels. Mechanical gated channels will open in response to touch, pressure, or vibration. So that will obviously be very important in the sensory neurons, because sound waves and stuff, that's pressure or vibration, for example. And then there are chemical gated channels, which open in response to neurotransmitters. They could also, of course, open in response to taste or smell molecules. And then voltage gated channels are going to open in response to the change in voltage, which is what we've seen with like sodium channels, potassium channels, and those calcium channels. The chemical gated channels that we've met so far were like the acetylcholine back in the muscle chapter. All right, so remember the resting membrane potential is negative 70 millivolts. It can vary. So some places you'll see it as negative 40 millivolts all the way up to negative 90 millivolts. So just remember that the average is a negative 70 millivolt resting membrane potential, but it can vary. And then there are two other potentials to mention here. One you recognize, you've seen the action potential before, but let's talk about graded potentials. These are those localized depolarizations, and this is going to be similar to like the end plate potential that was talked about in the muscle chapter. So graded potentials are localized depolarizations that occur along the dendrites in the cell body, and they will influence the excitation of a neuron at the axon hillock. So graded potentials being those localized depolarizations, if they are excitatory, they will bring the axon hillock towards threshold and will initiate an action potential. If they are inhibitory, they will take the axon hillock away from the threshold, and they will then prevent an action potential from occurring. So graded potentials are very important in the behavior of a neuron. So if the graded potential is excitatory enough that it reaches that threshold, an action potential will occur, and that's the depolarization of the neurolemma. And that neurolemma is the plasma membrane of a neuron. And this is what is known as the nerve impulse, that action potential. It travels all the way down the axon away from the cell body towards the axon terminal. And this is what results in the release of neurotransmitters. The cell membrane of a neuron, as I mentioned, is the neurolemma. And at rest, the neurolemma maintains a resting membrane potential, abbreviated RMP, of negative 70 millivolts. And the sodium-potassium pump maintains the RMP by actively exporting 3 sodiums out of the cell and importing 2 potassiums in. And this results in a higher concentration of sodium outside and a higher concentration of potassium inside the neurolemma. And because there's leakage channels, the sodium and potassium is always going to want to, move with its concentration gradient. So sodium is always going to leak back in, and then potassium is going to leak out. And there's actually more leakage channels for potassium than there are for sodium. So potassium leaks out at like a higher rate. And that is one of the reasons why it stays at like a negative 70. But the sodium-potassium pump is going to have to continuously kick the sodiums back out and bring potassiums back in to keep it at negative 70, because with the rate at which potassium leaks out, it would get more negative than negative 70 if it wasn't for the sodium-potassium pump. And that's what we've seen in like hyperpolarization. Remember that it can get all the way to like negative 90 millivolts or more, and that's because that potassium is going to continue to leak out. All right, so here is the illustration of leakage channels, and notice even in this illustration, it shows more potassium leakage channels. There's 3 potassium leakage channels shown in blue, and there's only like 1 sodium leakage channel that's shown in purple. And so sodium is going to be actively pumped out of the cell into the extracellular matrix there, the interstitial fluid. And the potassium is going to then be actively pumped back in. And so there'll be a higher concentration of potassium in the intracellular fluid or the cytosol. And the potassium is going to want to then naturally leak back out. And the sodium is going to naturally want to leak back in. And so the sodium-potassium pump is constantly pumping out 3 sodiums and bringing in 2 potassiums with each cycle. And that results in that negative 70 millivolts. You see that there's protein here. This is an anion, a negatively charged ion, that is going to be attracted to the potassiums, and that also plays a role in that negative charge that's on the interior of the plasma membrane. Notice also, before I move on, that ATP is involved with this. So this is another reason that ATP is so imperative to the body, because it not only provides like energy for muscle contraction, as we've seen, but it powers the sodium potassium pump. So it provides energy for the movement of this protein as it's pumping the sodiums out and the potassiums back in. Thus, the ATP is like directly involved in maintaining this resting membrane potential so that the neurons can conduct their action potentials, and so can the muscles. Voila. Let's revisit the 2 phases of the action potential, and that's the depolarization and the repolarization. So the depolarization is going to occur when sodium is flooding the axon. So voltage-gated sodium channels have opened, sodium is flooding into the axon, and that's going to spike the action potential up to a positive 30 millivolt. And then once positive 30 millivolt has been reached, the sodium channels are going to close and the potassium channels are going to open. And that's going to cause repolarization. So the interior of the neurolemma will return to the resting membrane state as potassium exits the axon because those voltage-gated potassium channels have been opened. And hyperpolarization will occur as the potassium continues to be expelled, because remember, we're losing more potassium than we would bring in sodium. So after that happens, the voltage-gated potassium channels will close, and we are going to rely on the leakage channels mentioned before, as well as the very important sodium-potassium pump to return to the original resting membrane potential. Let's see if this video will show. These are living neurons that are actually firing, and you can see that they are indeed electrical. So what the scientists have done is they have put like radioactive calcium or something in there, so that as the calcium, which is important in nerve impulse transmission, is moving through voltage-gated channels, you're seeing it light up. So remember, voltage, current, resistance, all of those properties that are found in the physics of electricity are occurring in these neurons. This is a illustration of the action potential, and I really like this one. It's a little different than what we've seen in the muscles because you're seeing graded potentials here taking us towards the threshold. And so these would be excitatory grading potentials that are going to be influenced by, the influx of sodium from those graded potentials down the dendrites and the cell body. And so once they reach that threshold, which is a negative 55 millivolts, then it will shoot up into the action potential. And the action potential is like all or none. It's either going to happen or not. And it has to hit that threshold in order to fire. Remember, like that gun fires when the trigger is pulled. And action potentials are always going to stop at that positive 30 millivolts. And as soon as the positive 30 millivolts is reached, then those sodium channels are going to close. And once those sodium channels close, then repolarization occurs as the voltage-gated potassium channels open. And as the voltage-gated potassium channels open, the potassium will leave the cell and bring us back towards that negative value. And eventually we're going to bypass the resting membrane potential and hyperpolarize. And then this is what will close the voltage-gated potassium channels. And then with the leakage channels and the sodium-potassium pumps, we will eventually reestablish the resting membrane potential. I apologize. My dog is annoyed that I'm lecturing and not playing with and giving him attention. He says, darn right. Bailey, hush. Okay, so let's look again. This is one you have seen. It actually follows the sodium and the potassium permeability. So this one was shown in the muscle chapter that shows that as we go towards threshold, the sodium permeability is increasing and sodium is going into the cell. That's going to bring us to that negative 55 millivolts. If it does, then sodium just floods in to the cell as voltage-gated sodium channels open, and then that depolarizes us to positive 30 millivolts. Once we've reached positive 30 millivolts, then the sodium channels are going to close, and notice that sodium permeability drastically decreases. Instead, now the potassium channels are opened, and we're going to see an increase in potassium permeability. But potassium is leaving the cell, and as it leaves the cell, it's going to bring us back to that negative value, and eventually we're going to hyperpolarize and bypass the resting state. At this time, the voltage-gated potassium channels close, and the leakage channels and sodium-potassium pump will reestablish the resting membrane potential. Right after a neuron fires, it undergoes a refractory period. The refractory period is the time after the start of an action potential when it is either impossible or difficult to initiate another action potential. There are two types of refractory period, the absolute refractory period and the relative refractory period. The absolute refractory period occurs first, and during this time, no stimulus can initiate another action potential no matter how strong or how fast it occurs. So this ensures that the propagation will always go towards the axon terminal. Then there's a relative refractory period. This occurs just after the absolute refractory period, and another action potential is possible if the stimulus is going to be strong enough, because you can see that the initial segment is slightly hyperpolarized and further from the threshold. So the minimum stimulus strength is now greater for the threshold to be reached. Basically, those graded potentials have to be just beating the axon hillock towards threshold, just shooting it with excitatory potentials. So this is the absolute refractory period here, and the relative refractory period here. So the absolute refractory period is in periods 3 and 4. So basically during depolarization and repolarization, there's no chance for another impulse to fire. But then in 5, the hyperpolarization stage, we are in our relative refractory period. So we can come back to threshold at this time, which makes sense. And then if another stimulus is strong enough to bring us from that hyperpolarized state to threshold, then we will get another firing very quickly. Once the action potential is initiated and propagation occurs, there are 3 kinds of conduction velocities. In a bare plasma membrane, we're going to lose the energy. It's going to diffuse out. Like, remember how I taught you that during muscle contraction, we lose energy as heat, and so we will lose energy in a bare plasma membrane, which is why myelin is so important. So the bare plasma membranes are going to occur in dendrites in the cell body, and that's why they're graded potentials, because they're going to kind of pitter out, but if then another one comes in, they can add up. All right, then B, in an unmyelinated axon, there are going to be voltage-gated sodium and potassium channels all along the axon membrane that will regenerate the action potential at each point, and so the voltage will not decay. Conduction, however, is slow because movements of ions and of the opening of gates and channel proteins takes time and must occur before voltage regeneration occurs. And there are unmyelinated axons that are found in certain areas of the nervous system. But then we're mostly going to see myelinated axons. And this is because myelin will keep current in the axons because, the electricity cannot be conducted through that myelin, so it has to stay in the axon. And so the voltage will not decay much. So action potentials are generated only in the nodes of Ranvier and thus appear to jump rapidly from node to node. And notice that in the nodes of Ranvier, those voltage and potassium and sodium channels are clustered. So that's going to be where the action potential is repropagated. All right, so let's go further into the graded potentials. They are small, short-lived changes in the resting membrane potential that occur on the dendrites and the cell body. They are established in response to inputs from presynaptic neurons, and they conduct short-lived currents. A large stimulus will open more channels, allowing more ions to flow. So let's go in and explain what presynaptic neurons are. Neurons will form unions between other neurons, muscles, and glands. In order for neurons to communicate with each other in the nervous system, they're going to be connected to other neurons. So it's that presynaptic neuron, the one that is connecting to the neuron being communicated with, that's going to be what is sending in the graded potentials. The postsynaptic neuron is going to respond to those graded potentials, either excitedly or inhibitory. Excitatory postsynaptic potentials will depolarize the membrane, and excitatory input comes in from a presynaptic neuron, and this opens chemically gated sodium channels, making the neurolemma less negative. So that's going to bring us towards threshold. Inhibitory postsynaptic potentials are going to hyperpolarize the membrane, and an inhibitory input will come from a presynaptic neuron. It will open chemically gated potassium or chloride channels. Notice that chloride is negative. So we're either going to leave potassium out of the neuron, so potassium will leave, or we're going to bring the negative chloride in, and both of those actions will make the neurolemma more negative. So neurons can receive both excitatory postsynaptic potentials and inhibitory postsynaptic potentials simultaneously. And notice that excitatory postsynaptic potential is abbreviated EPSP, and inhibitory postsynaptic potential is abbreviated IPSP. This shows the generation of an EPSP, an excitatory postsynaptic potential. So number one here, the neurotransmitter released from the presynaptic neuron binds to the postsynaptic neuron receptors, which are chemically gated cation or positively charged ion channels, causing them to open. And so the sodium, basically. So once the presynaptic neuron opens the sodium channels on the postsynaptic neuron, sodium flows into the postsynaptic neuron, exciting it, bringing it towards the threshold. Here's the generation of an inhibitory postsynaptic potential, an IPSP. So in step one here, the neurotransmitter released from the presynaptic neuron binds to postsynaptic neuron receptors, which are either chemically gated potassium channels or chemically gated chloride channels, and that causes them to open. So if the potassium channels are open, then potassium leaves. If the chloride channel opens, the chloride is an anion, a negatively charged ion, so chloride will start to flow in. And both of those are going to move the resting membrane potential away from threshold, so it's inhibitory. Summation adds the excitatory postsynaptic potentials and the inhibitory postsynaptic potentials together and determines if the postsynaptic neuron is excited or inhibited from firing. There are two types of summation, spatial summation and temporal summation. Spatial summation occurs when multiple presynaptic neurons send EPSPs and IPSPs from different areas at the same time. So it's coming at the same time, just from different areas on the neuron. Temporal summation occurs when one presynaptic neuron sends multiple EPSPs and IPSPs and or over a period of time. So it will hit it from one presynaptic neuron. The summation may or may not reach threshold. Typically, the threshold of the initial segment is negative 55 millivolts. If the threshold is reached, voltage-gated sodium channels will open and an action potential will be generated and then propagated down the axon away from the cell body. So here you see this neuron is totally synapsed with many different other neurons. I mean, it is in high communication with lots of other neurons in the area. And we see that neurons 1, 2, 3, and 4, and 5 are all in the green. So that's going to indicate that they are EPSPs. So they're going to be exciting the postsynaptic membrane. And then neurons 6, 7, 8, 9, and 10 are going to be in the red. And they are going to be shooting the inhibitory postsynaptic potentials, which will take that postsynaptic membrane away from threshold. And so the right side graphs are showing spatial summation at the top and temporal summation at the bottom. I'll read one of the examples like from each. So in the spatial summation here, you see that we've got P1, so that's neuron number 1 over here, and P2, neuron number 2, will both be shooting excitatory postsynaptic potentials to the neuron. And when they are added together, it's going to increase the value and bring them towards that threshold. And then in temporal summation, we're going to see that 2 excitatory postsynaptic potentials, both from P1, are coming at different times, but they're going to actually be added together and will help bring the neuron towards threshold as well. So spatial is multiple presynaptic neurons coming from different areas, and they'll be sending their impulses simultaneously. So that's spatial. Multiple neurons from other areas. Temporal is one neuron, and it is going to be sending multiple impulses in rapid succession. We have EPSP and IPSP cancellation. So you get an IPSP from a neuron and an EPSP from a neuron that hit at the same time. So this would be spatial, and it's going to cancel each other out. Then we have spatial summation, which is going to bring us closer to threshold, because we see that excitatory potentials from many neurons will bring us towards threshold. So those are both positives there. And then in temporal summation, this one is going to be that one neuron that's sending multiple impulses in rapid succession, bringing us towards threshold. In order to effectively communicate with other parts of the body, neurons will synapse with other neurons, muscles, and glands. So with other neurons, neurotransmitters can stimulate or inhibit other neurons. Within the muscles, neurotransmitters can excite or inhibit cardiac and smooth muscles. Neurotransmitters will always excite skeletal muscle. So when you see here that the neurotransmitters are exciting or inhibiting cardiac and smooth muscle, go back and recall that smooth muscle, glands, and cardiac muscle are part of the autonomic nervous system, which is parasympathetic and sympathetic. So if it's sympathetic, in this case, it's going to be exciting the cardiac. If it's parasympathetic, it's going to be inhibiting the cardiac. Smooth muscle is going to vary based on what the organ is. But that's what that is referring to there for the muscles. And then glands. Neurotransmitters will stimulate the release of endocrine and exocrine secretions. And these are going to include endocrine for the hormones, and then exocrine will be like digestive enzymes, sweat, saliva, tears, stuff like that. And that's going to also be at the discretion of, particularly the exocrine glands, will be at the discretion of the autonomic nervous system. So, sympathetic and parasympathetic. Here are the three types of synapses in illustrations. You see the neuron synapsing with another neuron. So neuron 1 would be the presynaptic neuron in this case, and neuron number 2 is the postsynaptic neuron. And so you can imagine that this presynaptic neuron is going to be sending either EPSPs or IPSPs to the postsynaptic membrane. Then we see the neuromuscular junctions. In this case, this is a somatic motor neuron, and it is innervating skeletal muscle fibers. And so it's going to be exciting the skeletal muscle fibers because it will be secreting acetylcholine, which then binds to receptors and excites the skeletal muscle. And then we see a neuroglandular synapse here, and this would be, either an endocrine or an exocrine gland. The chemical synapse will release neurotransmitters and is the most common type of synapse found. The features of the chemical synapses include the presynaptic neuron, the synaptic knob, which is also called like the axon terminal, synaptic vesicles, which are going to contain the neurotransmitters, and then when the neurotransmitters are released, they're going to diffuse across the synaptic cleft, where then they'll bind to chemical-gated receptors on the postsynaptic membrane. Neurotransmitters are the chemicals that transmit information within the nervous system. More than 100 neurotransmitters have been identified and organized into different groups. Neurotransmitters stimulate an excitatory or been talking too long. Neurotransmitters stimulate an excitatory or inhibitory response in the target cell. Most neurons make 2 or more and may release any one or all of them. And most neurotransmitters are released at different stimulation frequencies. That's how the neurons can determine what's going to be released. There are 4 main chemical classes of neurotransmitters. We've got acetylcholine, biogenic amines, amino acids, and neuropeptides. Acetylcholine has a structure that differs substantially from other neurotransmitters, and it will include the acetyl group and the choline group. Vinegar is acetic acid, and so it's thought that, drinking pickle juice and eating mustard and stuff will help ease muscle cramps, and one of the thoughts behind that is because of the acetic acid in the vinegar helping the acetylcholine to be produced. So anyway, research is out. Then we've got the biogenic amines, which is an amino acid that is slightly modified to synthesize this neurotransmitter, and they include catecholamines and indoleamines. Catecholamines are dopamines, epinephrines, and norepinephrines, and they are made from tyrosine. Indoleamines are serotonin, and they're made from histidine or tryptophan. And as you've heard, turkey is high in tryptophan, so that can, maybe release serotonin after a Thanksgiving meal. I know you've all heard that before, whether it's true or not. Amino acids will include common neurotransmitters glutamate, glycine, and GABA. And then the neuropeptides are going to be chains of amino acids that will include endorphins. So this is a nice illustration of certain groupings of the neurotransmitters. Here you can see the acetylcholine, and that one is the one that is going to help the skeletal muscles. You also see that it's a learning neurotransmitter, and acetylcholine therapy may help Alzheimer's patients as well, because acetylcholine does play a role in cholinergic neurons of the brain that will help to arouse the brain and aid in learning and memory. Then we saw the biogenic amines, so we see that there's like dopamines and epinephrines and norepinephrines, and then serotonin. So there's your adrenaline and noradrenaline, which is and the dopamine, right? And then the serotonin. So all of those up there, dopamine, epinephrine, serotonin, those are all going to be the biogenic amines. And we see that adrenaline is a fight or flight neurotransmitter, so sympathetic nervous system, produced in stressful or exciting situations. Increases heart rate and blood flow, leading to a physical boost and heightened awareness. Norepinephrine adrenaline, and you've heard of adrenaline and noradrenaline before as epinephrine and norepinephrine. So this is a concentration neurotransmitter and affects attention and responding actions in the brain and involved in the fight or flight response. Also contracts blood vessels, increasing blood flow. We see dopamine, that's the pleasure neurotransmitter, and it has feelings of pleasure and addiction. And movement and motivation, people repeat behaviors that lead to dopamine release. Serotonin is a mood neurotransmitter and contributes to the well-being and happiness, helps sleep cycle and digestive system regulation, and is affected by exercise and the exposure to light therapy. So then let's look at the amino acids. We've got glutamate, glycine, and GABA. And I believe that glutamate and GABA are the ones here on this illustration. So we'll look at GABA first. That's a calming neurotransmitter. It calms the firing nerves in the central nervous system. So high levels will improve focus, and low levels will cause anxiety. It also contributes to motor control and vision. And then glutamate, as you can see over here, is a memory neurotransmitter, and it is the most common brain neurotransmitter. It's involved in learning and memory and regulates development and also the creation of nerve contacts. And then lastly, we had the neuropeptide neurotransmitters, and we saw endorphins there for that. And they are the euphoria neurotransmitters that are released during exercise, excitement, sex, producing well-being and euphoria, reducing pain. And they have a biologically active section that is shown in this illustration. Once the neurotransmitters have done their job, the removal of neurotransmitters from the synaptic cleft can occur in several ways. There are enzymes that degrade the neurotransmitter, like we saw acetylcholinesterase back in the muscle chapter that degrades acetylcholine. Another thing that can happen is the reuptake of neurotransmitter into the presynaptic neuron, and that's what reuptake inhibitors prevent from happening, like with SSRIs. And then the diffusion away from the synapse and then it being engulfed by surrounding neuroglial cells. And I see the SSRIs right here with the drugs. Bailey, it's like literally the last slide, Bailey. Hold on. So some drugs have their effect by influencing transmitter removal. So SSRIs, or selective serotonin reuptake inhibitors, will treat depression by inhibiting the reuptake of serotonin, thus keeping it in the synapse and helping to aid in its mood-enhancing effects. And then we see galantamine hydrobromide, which is an acetylcholinesterase inhibitor that is used to treat Alzheimer's disease, kind of like I mentioned before. Acetylcholine can aid in the patient's learning and memory. And that's it. Bailey, we're done. You could go outside now. All right. Thanks, everybody, for listening.